Hi everyone, welcome to our weekly reading group where we have an author of a paper that we think is interesting, present a paper, and we discuss it together. So if you want to join these sessions yourself, then all the information is down in the description. It's just a Zoom link where you can hop on. And now we're talking with Jérémie Kalfon about his scPRINT paper. Let's go. Is Gabriel also, did he have some works with Federic Villani or this Villani guy who won some I think the Fields Medal for some optimal transport work.
Yeah, I think I don't know how well they know each other, but I think they come from the same, ENS is where like a lot of those very good mathematicians come from in France.
Okay. Um, Figali. No, that's not him. Anyway, let's get into our stuff for today. Then, yeah, do you just quickly want to introduce yourself and then let's get started.
Okay, yeah. So yeah, I'm Jérémie Kalfon. Before it's a quick introduction of myself. So I started a PhD actually a year ago now, and before that I was in the field. So I worked at the Broad Institute in the project called DepMap for a few years. I was a computational biologist there. Then I worked in a few startups back in France, in Paris, and after some time I decided to launch myself into this endeavor of doing a PhD. So with some delay, but in the lab of Laura Contini and Gabriel Perret. So it's kind of a joint PhD between those two labs. One that is more like applied math, and the other one is more like computational biology. Cool. And so, this first project is called scPRINT, and it's really a transcriptomic foundation model that does, many things, amongst other inferring molecular interactions. And so, to introduce, I just want to explain a bit the main ideas behind that. So, in cellular biology, there is the kind of this big goal of being able to have a model of the cell, right? And recently you had this idea of having an AI virtual cell. And then the idea is if you are able to model the cell really well, you can predict the effect of perturbations and drugs in silico, and so speed up a lot the development of, for example, therapeutics. Um, and that's kind of the recent idea, but before there was also this notion of having a model of the cell that was maybe a much simpler, which is having gene networks. And a lot of tools are gene regulatory networks in France. And the idea is that you have a bunch of cells, if you do single cell RNA-seq, that you will measure, and that will be basically your matrix of n genes by m cells. And you want to have a way to transform that or to generate from this a graph of interaction between your genes. And that would be kind of your coarse view of a gene, gene networks and maybe how the cell, things in the cells interact with each other. And so often you call gene regulatory networks, those networks, because you only look at TF to gene connections. And transcription factor, TF, they are those genes that are known to have an important role in the regulation of the expression of the genes. And the parallel I want to draw is this is a picture of how a cell works. And often it's done with like very simple models, this gene regulatory network in France, like correlation-based models or random forests for the most famous ones. But what happens in the cell is not that, right? It's a very complex things of billions of micromolecules that interact with each other. But both of like this is a model of the cell, and this AI virtual cell is also kind of having a model of the cell, making it maybe more complex, but kind of the idea behind that is just kind of the same. Although now it's a black box, and this one is maybe the white box version. Okay, so to talk more about those kind of these AI cells or AI virtual cells, this is based on those new, not so new now, large language models, transformer models, where before I don't know how if you're used to neural networks, they are basically the same thing, except you could think of instead of working on vectors that represent maybe, for example, an expression of the cell, here they will be working on matrices, where each of your input elements will be themselves, instead of being scalar values, they'll be vectors. And so your transformer will basically update your matrices across its layer. And oftentimes you have to find a way to encode your input, for example, language or something else, into these matrices and decode them back. And something that has been known in the field from large language model, but also this protein language model that are trained on the amino acid sequence of the protein, is that within those models, you have these attention matrices that kind of encode the relationship between all of those input elements. And these attention matrices, actually, they contain some structure. And you can, from those extract the relationship between words in your sentence, but also the relationship between amino acids. It's called the contact map of your proteins, and that contains some of the structural information of your protein. And those models, they kind of learn that from scratch. They're not trained specifically to have this, just they have these simple pre-training procedures that we'll talk to you about in a moment. And so they actually have been applied in single cell data. You might have heard of those models like scGPT and GeneFormer. And thanks to big databases now, like the Cell by Gene database from Zuckerberg Institute, you have like more than 50 million cells that have been gathered, 1,000 data sets. And here you have a small picture of that where each point is you map a cell in this very large snapshot of the UMAP. And the idea is if you train those models on those millions of cells, you do this pre-training, they might be able to learn something very fundamental about the cell. And then you can fine-tune them on any task of cellular biology, and they should be able to be really good on that. At least that's the main idea. Okay, and so, okay, same slide. To talk just quickly about how those models are, how do they work? GeneFormer and scGPT before I start talking about SPRINT itself. Those models, they work on this cell by gene matrix, right? And if you train the model, you'll generate some mini batch of a set of cells. And these vectors that represent the genes that are expressed in those cells are really, really large. They can be 20,000 genes and sometimes even more. And the things are mostly empty, right? You have these notions of dropout, where lots of those, because of the way we sequence those cells, we have a lot of noise, and we are not able to sequence or to analyze all of the genes that are expressed in those cells, in most single cell RNA-seq data sets at least. And so the way these models work actually is they cannot have a huge context. They cannot work on like 10,000 or 20,000 genes at the same time. So they will reduce their input, and they will just say, we only look at the genes that are expressed. So if you have this here, the set of genes that are expressed in blue, they will only consider expressed genes. The rest, they will use padding, and they will stop at around 2,000 genes that are expressed. And they will only look at those 2,000 genes. And then they are trained using simple masking methods. And the last point I have to mention is how do you encode this information into these matrices for the model? So the idea is you have to use an embedding of the gene. Right? So you have to convert this kind of gene ID, the notion that this gene is MYC or this gene is something else. You have to convert that into a vector for the model to understand it. And so both of these models are doing the same thing, actually. They are learning and embedding a representation of the gene ID. And then you have to encode also how this gene is expressed in this one cell. Okay, so that's where scGPT and GeneFormer are doing actually something a bit different. GeneFormer is using kind of the ranked expression of the genes. So they take the most expressed genes in the cell, and that will be ranked number 1. The second most expressed gene will be ranked number 2, and so on. Okay, and that's just a way to encode your expression vector, to say. And the positional encoding is really a famous way to encode position in text, and here it's being reused to encode kind of expression. And for scGPT, the way it works is that you will bin your expression. So you will just say between 0 and 5, it's just bin into one. Basically, one embedding will be learned for anything expressed between 0 and 5, another one with for anything expressed between 5 and 10, and again, and so on. And so there's just two different ways to encode expression, and it's really the same way to encode the gene idea. So you have to learn this embedding during training, and actually that's a very important part of the model's weight most of the time, because you have 20,000 genes from which we have to learn embeddings. Some genes you will see often, and some genes you won't be seeing often, and so you won't be able to learn a good embedding of those genes. Cool. And so the last point I have to mention is that within those models, and that's what we were interested in, looking at those, that within those papers, there's been mention of gene networks and ideas of gene networks inference. And I won't talk about the way the scGPT does it, because I find it a bit strange, but you can go see that in their paper. But for GeneFormer, at least, there was this idea of doing an in silico gene deletion, where you just remove a gene from your input, and you just see, compared to not removing the gene, how this is changing kind of the output of your model, the output gene embeddings. And you're thinking that if the output gene embeddings are changing a lot when you remove that gene, it must mean that that gene has a strong impact on those genes, maybe expression. And that was shown in a few cases for some transcription factors in some cells, but it seemed to be performing interestingly and even to get a direct versus indirect effect, which is something actually quite hard to get solely from the data. And so this is all good and well, but maybe you've heard of this as well, but there's been a few for now preprints that have kind of criticized these models. And I will add also some of my own criticism. But one of the notions is that actually in some context, pre-training doesn't help the model's performance at all. Meaning, if you fine-tune the model from random weights, or if you fine-tune it from a pre-trained model, you won't really change your performance in some tasks. Here it's for predicting perturbations, for example. The other thing is that in most benchmarks, they don't outperform the state-of-the-art tools. So you could say, like, why would I use these models if I can get something much simpler, much faster to run that works better? And some of my own criticism is actually, for these models, we actually have almost no information. You can't reproduce the pre-training. What was released is just the train parameters and a way to do inference with the model. Some fine-tuning, but not how to retrain the model from scratch. And when you look at the pre-training itself, it seems that there was no weighted random sampling, for example. So the issue is that in the big data sets of cell by gene, you have many data sets that are actually exactly the same or very similar from the same tissue. And on those, you also have you sequence many times the same cells. And some rare cells that have a lot of variability, you have just a few of those. So you really want to do weighted random sampling, and some paper have mentioned this issue as well. Okay. So I think that gets to kind of the idea of this project. The first one was to ask the question, how much large cell models, kind of the name I give to those large language models apply to single cell data, how much do they have really learned about the cell? Can we assess this in some different ways? Can we and maybe can we assess that using these notions of gene networks and attention matrices? And can we maybe improve those large cell models using the framework of gene networks and gene regulatory networks? Okay, and so that kind of get this idea of sprints. We ended up having to build a new model from scratch because, well, not completely from scratch, but because there was, as I mentioned, a lot of issues with the previous one, and it was really hard to get at how those models were pre-trained and so on. And so that's what I will present also quickly, so like sprint. And this is a really coarse overview of what it is, and I will go through the different steps. And you'll see a bit how we get data, single cell data, how we encode it, how we train those kinds of models, and the different possible ideas to do that. And I hope also I'll make some notion of why transformer model might be actually very well applied to these kinds of data sets. Okay, I will start with the basic, the data loading mechanism. So I mentioned there was no data loader for this, and the first thing we had to build, and I did that actually with a group of good people at LaminDB, which is a company, but also a toolkit for working with large single cell data sets. And so I released something called SC Data Loader that you can use now, which works on top of LaminDB, that allows you to, for example, download all of cell by gene pretty easily, pre-process it, and do weighted random sampling on that in a way that doesn't really bottleneck your pre-training performance. And so this way, if you have rare cells or cells for which you want to sample more, you are able to do this very fast. And this is how Sprint has been trained. And I believe it's also the first pre-training that has been done this way. Okay, so once you're able to load your expression profiles, how do you encode it? I mentioned a bit how scGPT and GeneFormer do it. For Sprint, we do it a bit differently. And I believe the first paper that talked about this is the universal cell embedding, which is another foundational model of the cell. And here, we don't actually learn all of those gene embedding that we have to give as an input to the model. We don't learn them from scratch. What we do is we take the most common protein product of those genes. We use the amino acid sequence and we give them to a pre-trained protein language model. And from those, we extract an embedding of those proteins, hoping that not only we reduce completely the number of weights that are having to be pre-trained, but also we encode a lot of information about the protein. For example, some information about maybe its location, its ontology, and that might be useful for the model to learn gene networks. The other thing that we encode, obviously, is the expression. And here we do it also a bit differently, where we use just simply an MLP that takes the scalar value of the gene expression that you have and converts it into a vector. And the idea for that is, we don't apply any like ideas of how we should encode. We had a lot of questions of how do you encode expression, and by just using a two-layer MLP, you basically let the model learn it from scratch. And maybe that's the best way to do it, because it will decide if the bin size should be this or that, or if genes that are highly expressed should be really encoded differently or not. And the last part that we added actually also is an encoding of the gene's location in the genome. And we did this because there's this idea that genes that are very close together in the genomes are being regulated by similar regulatory regions. So again, that might help the model learn those regulatory regions and learn some networks afterward. Okay, and so what?
Denise is asking, how do you harmonize expression counts between cells and sequencing platforms?
It's a good question. So I think that is even actually a problem behind the way GeneFormer does its encoding. I don't think it's well harmonized with the way they do things. At least the first version of GeneFormer. And in our case, we don't do much. We just do a log transformation of the data. So we take the raw counts and just log transform, and that's it. And then we let the model learn from that.
Okay, and do you always have one chromosome or multiple chromosomes and multiple? Then how do you account for positions on the different chromosomes? They don't have an intrinsic order, right?
Well, it will be from chromosome 1 to chromosome 21, but what's important is really the close relationship. So it's just for the model, what we hope it learns is really that the sets of genes are close together and the rest are far away. And so maybe you don't really care about as much, if chromosome 1 or chromosome 2 should be close or not. Maybe if that was your question.
Yeah, but like the thing is now, right, if you just concatenate them, for example, then the end of chromosome 1 will be close to the beginning of chromosome 2, while it isn't in reality. And I guess the question was mostly about, do you take care of that somehow? Do you use some positional encoding to make sure that there is now no relationship, like there's no position relation, like you could have a permutation invariant positional encoding between different chromosomes, while inside of a chromosome it is you still have positional relations.
So actually, I don't exactly remember the details of this exact part of the implementation, because now it's been a while. But you can check on the code, and I'm sure at least that it wasn't that complex. I didn't introduce those kinds of invariances in the encoding of the location. But that would be a really interesting update actually. The other part is how do you you have this list of genes, and again, SPRINT cannot work on 20,000 genes at once. But most of the time you only have 2,000 or 3,000 genes that are expressed at the same time in one cell because of this Durand issue. Right? Although the cell likely has, we know that the cell has around 12,000 or more genes expressed, we only have those 2,000 or 3,000 that we can assess. So what we do is we select, basically we use a set of randomly selected expressed genes. That's how we give our input to the model. So any genes expressed, we randomly select them. If you have less than 2,000 genes, because that's the input context of SPRINT, less than 2,000 genes that are expressed in the cell, what we do here is we don't do padding, but we add randomly selected unexpressed genes. And you'll see why this will be kind of important and interesting to do for the pre-training step also. Okay. So I don't talk about the transformer itself. Really, it's a kind of regular bidirectional attention, self-attention transformer with flash attention too, which allows it to work quite fast actually. And also, if you look at the paper, we have quite fast training. It's not taking like a month on 20 GPUs. It's faster than this. And I talk now about how do you decode those embeddings again to pass them into this vector of gene expression at the output. And so the way the other models did it in the other foundation models is they do some non-parametric loss where they use mean squared error and they only have a multi-layer perceptron that predicts the scalar value and you compare it to your true count values. However, in the field, models like scVI and scRCs, they've shown that something else that works really well is to use a zero inflated negative binomial distribution, which maps really well to your data. So this is a decision where you basically say, I know my data looks like a zero inflated negative binomial. Can I just predict the parameters of those distributions? And then, which is what scPRINT does, basically it's using a zero inflated negative binomial loss, where it predicts the parameters, 3 parameters, basically the mean, the dispersion, and the likelihood that this gene is expressed, but you haven't sampled it because of noise and of dropout, which is this dropout term, which is a zero inflation. And then your loss is basically the negative log likelihood of your parameters given the data. Okay, so that's for basically how you input and how you output data. But now, kind of the most important part of those models is actually how do you train them and what is your loss. And here we also tried something a bit different with scPRINT, where we don't have just one loss and we don't do masking, but we do a multi-loss pre-training or multitask pre-training, like other language models have done before. And I will present them, to you one by one. And the first one is we don't do masking, we do something called denoising. And you might have heard of denoising for like diffusion denoising models on images. So here it's not a diffusion model, it's just doing denoising. And this notion is like the data, RNA-seq data is, as I mentioned multiple times, quite noisy. And we want the model to be able to know about this and to model the noise process. And the noise process here is a sampling noise, right? So we sample some counts, we sample some reads, and we don't sample some other. And so the denoising here is not, we don't add noise using a Gaussian process. We add noise using this kind of Poisson process or zero inflated Poisson, where basically we decrease the counts to, if you want to add 70% noise, we'll make it so that the input expression profile will have 70% less counts, or the depth will be 70% lower. And you have a probability of just decreasing the number of counts for each gene using a Poisson, and a probability of just dropping out entirely this gene. And now you see why that might be useful to have padded the input data with kind of non-expressed genes, right? Because now you let the model learn about genes that are non-expressed because they are really not expressed in the cell, and the ones that are just not here, but they are actually likely expressed, and you want the model to be able to denoise them and recover their expression. So it gets allows the model to do this thing basically. So that's the first step and the first training procedures. And the idea is also, this is a way to have the model be able to predict that gene's precise expression using the expression of the other genes in its input. And that's how also you hope the model can have learned some better gene networks instead of just masking 10, 15% of the genes, which is also just not a lot. And before I talk about the other pre-training tasks, let's just look at what this allows scPRINT to do as kind of a zero-shot behavior, meaning you don't have to fine-tune it in any way. And denoising is an important task in cellular biology. And so here what I'll show you is results of scPRINT when you take I add noise to a data set. I ask scPRINT to denoise it, and I look at how much it improves the correlation of the output expression before and after having applied the scPRINT model and denoising. And for that, there is two very famous, I'd say, state-of-the-art models that are used in the field, KNN smoothing 2 and MAGIC. And those are using basically the nearest neighboring cells by using a KNN algorithm, and using those information to predict the denoised expression profile of those cells. Sprint doesn't do that. It takes one cell at a time and denoise them from scratch. And actually, Sprint doesn't outperform here in this context, the state-of-the-art tools on the dataset. However, what happens is because it's not using the neighboring cells information, when you look at rare cells in your datasets, the one that are these rare cell types when you only have sequenced maybe 10 or 20 cells, interestingly here, and actually I think it makes sense, Sprint doesn't really degrade in performance, but the other models do, because they're not able to use as many nearest neighbors. And there actually Sprint cannot perform state-of-the-art models in this task. Okay, so the other part, which is also kind of an important task in cellular biology and single cell data, is to generate a cell embedding that you hope kind of represents and compress the expression of your cell or your cell information into this one vector. Okay, and so this is part of our pre-training task to be able to generate a cell embedding. And there was a task that actually that scGPT introduced, which is this, or a way to learn this, which is this gene cell product. Where what you have is you take your gene ID embedding that just encodes which gene we are talking about, and your output cell embedding, and you make sure that the dot product of those two can recreate your expression of gene I in cell J. And this is how you make sure that your cell embedding contains all of the expression information of your gene's expression in that cell. What we presented with Sprint is another possible approach where you mix the idea of an autoencoder and a transformer model. So basically here you will reuse the same Sprint model bidirectional encoder to kind of decode from the cell embedding the expression profile. So the model takes as input initially the expression profile and the gene IDs, right? And it will output its output including a cell embedding. And it's the way we kind of use it as a decoding mechanism is that now we don't give the model any information about gene expression. We just give this cell embedding CJ and the gene IDs, and we ask it to reproduce to be able to generate the expression of that cell. So you use twice as much compute basically, but what we've shown is this this performs better. It makes sense given the amount of compute you use, and it's able to recreate a better expression profile than just the gene cell product mechanism. Okay, and so this is part of the pre-training, right? And before I talk about how this cell embedding compares to maybe to other models, what I have to talk about is kind of the last task, which is also not common, and that's one thing that we do is we don't generate just one cell embedding, one vector per cell. We generate multiple cell embedding for one cell, and each of the cell embedding actually encode the idea that they should encode a facet of the cell. A reason for that is we have this notion of batch effect in single cell data, where, for example, the sequencer will introduce a lot of batch effect in your embedding. And what we want to do is somehow to let the model separate those different kind of factors that influence the cell expression into different embeddings, and maybe have one that just represents the cell state and less the sequencer. And so we introduce classification where for each of the cell embedding, we'll have a classifier predict one of those different things like cell type, disease, sex, sequencer, and so on. And this is done as part of the pre-training. So it's not something that happens often, like in Vision Transformer, you won't see that at all, and that's because you don't have labels for images most of the times. However, in our context, for single cell data, you have, in cell by gene, for example, you have labels for all of the cells, for at least cell type, dataset, sequencer, and disease. And so you can use those for pre-training. And again, the hope is, given you have noise, those labels should be quite useful for the model to learn about maybe the marker genes or the important parameters and genes to be looking at. And so one thing that we had to solve, however, to be able to do that, is that those labels are not always the same. So sometimes you can see something like neuron, and sometimes you can have something like mitral neuron. And those two things are, the labels, a mitral neuron is a neuron, right? So you have this kind of hierarchy of coarseness of labels you have to take into account. And so we designed this kind of classifier that we call a hierarchical classifier that allows you to work on this kind of data. So this data is called, it's kind of defined in an ontology, which you have access to. And so what we have is that we let the model, with less print or the classifiers, predict only the most, the leftmost labels, so the most precise labels possible. Okay, so that also reduces a lot the number of labels to predict. And whenever in the input, in the training data, we have something, for example, like neuron, which is a really coarse labels, we'll look at and we'll aggregate all of the model precision, all of the model predictions for the label that are part of the neuron category using a max, basically, of all of those logits. And that will be the model's prediction for the neuron category. And that allows you to make prediction at different level of coarseness in the model. And actually, also, we show that this works really well in practice, and the model achieves good performance in predicting cell type. And so, don't, we'll look at the results, so don't just take my word for it. And the first one is just how is the model able to correct some batch effects. For example, we'll just take the cell embedding that represents cell type and look at how it encodes the cell embedding and how it removes the sequencer information. And in this context, you have a pretty good benchmark that exists called from Open Problems benchmarks, and a package called scAB that basically gives you a score for this. Unfortunately, I don't think the previous foundation models were really benchmarked on this, like the same benchmark that every other models in single cell biology were assessed. And so that's what we did here. So we took the same data sets, so we can compare across every possible models, and we also used GeneFormer v2 and scGPT. The thing though is that we have not fine-tuned those models. So this is a raw performance, and scPRINT also is not fine-tuned here. It's zero-shot performance out of the box. And so what you can see first is that in this context, scPRINT outperforms GeneFormer v2 and scGPT, and it is actually removing some batch effect compared to just a simple PCA here, unintegrated, but it's clearly not outperforming the state-of-the-art cellxgene model. And actually, we just recently were able to run another model called scFoundation, and we've seen that it works very similar to scPRINT, tiny bit better. But one thing to know is that there is a big difference between those models here and those ones, because the models here, they don't have any information about the batch label, so they don't know which of the single cell data you get is from a different sequencer or not. They just get raw sequencing data and predict an embedding. And also they don't train on the test data set. These other models, you have to train them on your test data, you have a split, and that is also kind of a big difference that helps them a lot. So I think it's kind of an unfair comparison. But here what you can see is still that the model are able to perform. And if you do fine-tuning, obviously you'll be able to perform as you've seen in scGPT and GeneFormer's paper. So quite well. So that's one score, and the other one that exists is how you're able to preserve biological variations. And actually that's where scFoundation outperforms a bit scPRINT, and in the batch effect correction, scPRINT outperforms a bit scFoundation. And there you see that those models here, just in zero shot, scFoundation and scPRINT, they are performing similar than MNN. Just to say it's not too bad, especially in zero shot on a dataset that you've never seen before, and using and not even using the batch labels as inputs. Okay, and so the second task here is predicting cell labels. So we look at cell type prediction, but scPRINT can perform many other type of prediction, like as I mentioned, sex, sequencer, disease, ethnicity, zero shot, without having to being fine-tuned on anything, just from raw sequencing data again. And again here, scPRINT does not perform better than the state-of-the-art tools like machine learning models like logistic regression and XGBoost. But that makes a lot of sense again, because again here scPRINT does not train on these test datasets. And what happens is that logistic regression, XGBoost, they only predict over like 10 or 20 or 12 labels. Here it's 12 labels actually of these pancreas datasets. scPRINT here, it makes a prediction over 300 labels, possible cell labels. So it predicts over every possible cell type. So it's again completely unfair to compare those two. But even then, there was a few cell labels that were very, very precise. Pancreatic A cells, islet A cells, D cells, B cells, that scPRINT was not performing that well on. And just if you make the labels a bit more coarse and you reduce that to 7 or 6 labels, I believe, there you see that scPRINT performance jumps quite much higher, this, on the coarse labels, and the performance of XGBoost and logistic regression actually decrease. And here the reason is that by doing this, I kind of artificially make a very small set of rare cells, basically. And XGBoost logistic regression, they will have a bias towards the most common cell labels if you don't introduce any type of regularization. And so that's why it kind of goes to show that SPRINT can predict very rare cell types really well. And again, that's kind of what you care about the most. And so a better comparison is to compare SPRINT to something like CellTypist. And CellTypist is again also a model that just use cell markers genes, so just based on the literature, to be able to predict if this one cell is which cell type. And this is what is being used by labelers to be able to label their cells actually from scratch when you don't have any information about which cell type we're talking about. And there you can see that SPRINT actually outperforms CellTypist on these data sets. Cool. So I said that SPRINT was doing gene network inference, and I'll go to it now. But why did I do all of those benchmarks?
Sorry,
Well,
before that, maybe what do you think are the bottlenecks limiting the development of accurate generalizable cell models?
well, there is quite a few. Like if you go to, for example, just cell label, right? I think there is a point where those prediction accuracies from logistic regression are a bit unrealistic. Like it works well on this one data set where you have good labels and sometimes even not that good labels, I believe. And you just train it on this data set where you already have labels and then make predictions. But SPRINT has been trained on all possible labels in cellxgene, and talking to I don't remember his name, but the PhD student that had made a CTAB, an issue here is just the quality of cell labels, for example, and just the quality of data. And so that goes for me to say that the quality of input data is still a big issue. Like I mentioned, for single cell data, you only sample 2,000 or 1,500 of genes that are expressed in not all of your cells. A lot of them you discard because you only have 500, 200 genes expressed or being sampled. Whereas what really happens in the cell is you have 15,000 genes expressed. And so just the input data that you have is not that good. And if you compare it to protein language models, the sequencing information that you have for your protein is much better quality, and you have much less kind of noise and error than you have for single cell data. So I'd say that's one thing. And the converse of that is that you need much more data, right? Given the noise in your data, maybe you want to aggregate, take the average of a group of cells that are very similar to create those meta cells. If you start to do this, you realize that you don't have that much data as an input. So 50 million cells seems like a lot, and it's a lot. It's a few terabytes, but in the end, it's not that much to train your model. So that's the first thing, and then it just requires a lot more work. Like I did sprints myself. It took me a lot of time, and there is like I have a really long list of things to improve sprint, to do, like for example this task much better, and every other task I mentioned could be improved dramatically just by spending more time thinking about those issues.
Okay, nice.
Cool. And so, okay, so I mentioned those tasks first because I think it's important to be able to benchmark those models and those claims. And also because, you'll see now that gene network inference is not an easy thing. There is a reason why models don't do that well on that. We don't have a good ground truth. And so the idea is that a model with a good gene network should be performing well at most of those kind of known single cell biology tasks. Or like if you have a model that, from the same set of weights can do all of those very different single cell biology tasks quite well, you must have learned something that kind of makes sense about the cell, right? You must have learned gene connections that are not completely spurious. And that might be a way to assess kind of what's going on within those models, and might be a way to maybe validate your internal gene networks representation, given that the ground truth that we have right now for cells are really poor. But still, they're interesting, and I think we'll have to look at them to have a sense also at, are the models that we've built, those large foundation models, did they learn something meaningful about the cells, and can we find within their network, within those foundation models, can we find gene networks that make sense, that reproduce biology? So, okay, I talk about SPRINT. I say it takes raw cell expression profile as input, and it can predict cell labels, denoise expression, a deconvolved embedding from scratch, zero shot, without having to fine-tune the model. Can be fine-tuned, but those are kind of zero shot. And it also does gene network inference. So I believe any of those models could do gene network inference, those transformer models, because now you've guessed it, how we do it, it's using the attention matrices of those models, and here of sprints. And so similarly to what ESM2 showed, or basically kind of reusing the same idea, didn't invent anything new. But the one issue that you have to think about is in one model, you don't have just one attention matrix, you have many heads. For SPRINT, for example, you can have up to 200 heads. So how do you aggregate this information to get at connections between genes that are scalar values and not big vectors? You could use, but you'd have issue on kind of comparing them and benchmarking that. And so the main idea is you would do mean pooling. You would just take the mean of all of those attention matrices. But what the literature had shown before in language and in proteins is that a lot of those attention matrices actually contain some noise, and you might want to discard them, or some of them specialize to specific things. And so one thing you can do to improve your kind of network is to take a bit of your ground truth and to see how it correlates to your different heads. And if it correlates well, or if your heads are able to predict this bit of ground truth quite well, you just select them. And then you have a set of heads that seem to match your ground truth, and you can take the average of those. So you don't train a classifier on top really, but you just use the information of something like a classifier to aggregate a set of heads that you think makes sense. And here, this is also one thing that we can do with sprints, and for which we'll show the results as well. Okay, and so the first benchmark that we show is on a dataset of gene network called OmniPath, which is, I think, the biggest one, coming mainly from the literature, but also from experiments. Some of the connections are really good, some of them are not that good, and also from known kind of information about kind of networks like cell markers, transcription factors, and so on. And I've developed this package called BenGRN that allows you to basically benchmark a gene network to all of those ground truth datasets. And the way we do it is that I'll take 3 test datasets that SPRINT hasn't seen again, and I'll take a set of cells from the same cell type, and I'll generate a network from those. And then one question you can have is, well, SPRINT generates, takes one cell at a time. So how do you aggregate over many cells your gene network? And so, again, you could think of just taking the average of all of your networks for the set of cells that you give. Here we propose something a bit different, where if you know about how you compute an attention matrix, instead of taking the average of the attention matrices themselves, we take the average of all of the Qs and the Ks, that are the keys and the queries that allows you to compute your attention matrices over those cells. So when we do the forward passes, we store the Qs and the Ks, we take the averages of those, and in a way that allows kind of all of the keys and the queries to attend to every other keys and queries across the cells. And it also kind of denoises a bit the attention matrices. And we've seen that it works quite well in many instances. Some of them maybe a bit less, but most of the time, this is actually quite a very good solution. And it allows you to generate a genome-wide gene network, which is something that most models cannot do, like a gene network of 20,000 by 20,000 elements. Okay, so let's look at the results. The state of the art here, it's known for a while. It's the model that is very simple called Gene3, which is based on random forest. And here I also show you GeneFormer v2, scGPT, and SPRINT. And basically there is two. First thing to say is like I don't show you AUPRC. I show you the EPR metric, which is a bit different. It's basically the odds ratio value, where 1 would be completely random predictions compared to the ground truth, 2 would be twice as better as random, and so on. And this is compared to the big omnipath ground truth. And if I don't show you the AUPRC, it's just because it would be super low. Like it's like 0.00 something for all of those models. And that's mainly because omnipath, as the ground truth, contains a lot of false negatives. Just for some genes, we know a lot about their connections. For most of the genes, we don't know almost anything, and we just know maybe one connections or none. And so that's one of the reasons why also the EPRC is that low. And most of the time in the field of gene network inference, people use the EPR matrix. And there's two assessments. I show you the EPR on all possible connections or just on connection that relate to TF2 genes. So kind of gene regulatory networks type connections. And there, SPRINT, just the base version of SPRINT, kind of outperform GeneFormer v2 and scGPT across more than 20 cells, many data sets. And also even outperform DeepSem on the TF-gene, so gene regulatory network only version. So that's interesting, but Gene3 still outperforms everything else. However, if you start to, here on one cell, on one cell type, we've just decided to select a set of heads in SPRINT that seem to correlate with the ground truth. And we reuse the same set of heads across every other cell type. Then we have this SPRINT with omnipath heads only. So a subset of heads where you kind of remove the noise of which heads you're using. And there, you see that SPRINT just outperforms now every other model on omnipath. And we are able basically to find a set of heads that correlate a lot to what we know about gene networks and how gene relates to each other. So just that is interesting. It's just not to say that SPRINT should be the one model to use in this context, but just to say that actually what those models learn are things that make sense. And if you select
Sorry,
the head,
yes, can I please ask a question? Sorry for interrupting. I got
no, go
a
ahead.
bit of luck. Yeah, my question is, what kind of gene networks we're talking here? Because gene networks can be of many different modalities, and they also can come at the resolution of a single cell or at the resolution of a cell type. And what I'm kind of concerned about is that by marginalizing your keys and queries over cells that I assume you think are coming from the same cell type,
Yeah.
it can still remove variation between individual cells. I mean, it's obviously still useful, but how you come up with like a possibility of destroying some important data.
Well, so actually you'll see some of answer to your questions in next slides. But so you're right, I haven't mentioned omnipath is not cell type specific. It's just known connection between genes, and most of them are TF gene connections. I'd say maybe 50%, and the rest are maybe from PPI connections. You have some RNA connections. You have a lot of type of relationships, and here we don't subset it to any types, and it would be hard to kind of get those types of benchmarks on specific subtypes of like networks, gene networks, because there's not that much ground truth.
I see. I just saw at some of your images that for different clusters you kind of extract a network. So I thought that you compared to some kind of a ground truth for this cell type. But
Yes.
yeah.
So in the next slide, I'll show you the cell type specific ground truth. But bear in mind that all of those networks that SPRINT predicts are cell type specific. I run network on one specific cell type every time. Okay, so those networks for all of those models, they are cell type specific. And unfortunately, at least for this benchmark, we only compare it to a non-cell type specific ground truth. And the question we ask is just some of those connections at least should be like we should see something that is not random, right? So as more non-random it is, the better it is. But here it's just 3 times better than random. It's not amazing performance still.
Okay, thank you.
And actually, what you'll see some other results. So here it's not really omnipath, but we can also ask questions about, do my network make sense based on, for example, I would expect a gene network to have an enrichment, so have more connections for genes that are transcription factors. And so this is what we're looking at, is how enriched the network is for transcription factors. Or in green, you have how enriched the network is for cell type markers of the cell types for which the network was generated. So here you're basically asking, is my gene networks at least a bit cell type specific? And you see the results here where scGPT actually performs really well. Interestingly, or I mean better than the rest. And you see the results are also very different across the board. So GeneFormer performs poorly, SPRINT perform on average quite well, SPRINT with omnipasses heads as well. And a model I haven't talked about is SPRINT genome, where here it's the same thing as SPRINT, except I just ask it to generate a genome-wide gene network. Instead of having a network that is the 5,000 genes by 5,000 genes, like all of the other models can do at most. Here I can use SPRINT to generate a genome-wide network, and you can see that there you have a huge transcription factor enrichment. And if you look at something else, which is how much the connections of your transcription factors are enriched for known TF targets, so transcription factor targets from the ENCODE datasets, you also see that here SPRINT genome perform really well. And the main reason I think is mostly just because you have more genes, and so you have more statistics, and you can get a p-value for those, which you couldn't get before. So that's why SPRINT genome performed that well. But it also shows that you recover a lot of the ENCODE TF target connections from those models, and again they're not random. Like you have clearly learned something. Like except scGPT performs random on this, but still it's better than random, and SPRINT here outperform every other model on this interesting task.
Yes, though interestingly, transcription factors are also combinatorial in nature, meaning that enrichment of transcription factors for like a larger subtype, if you kind of go in the cell subtypes that are smaller, they can be non-linearly dependent, meaning that yes, we can have motifs connecting a given transcription factor to a lot of genes, but in some specific cell types, or like variations of cell types, those may not be correct actually. Those can be too broad. But
Sure.
okay.
And that's why we're looking at enrichment, and that's why also we are not expecting to get amazing result. Like if I see something that gets 100% prediction at even like cell type marker prediction, or this, I would be as most people, I guess, I would be very skeptical of the results. And that's the same thing for the OmniPath EPR values. Like if you get an AUPRC of 0.5, what you get in some papers that assess gene network inference, you're like, I mean, that's really weird. Like I wouldn't expect that, that high of a quality for a cell type specific, at least network. And so the other types of ground truth that we've used, I won't show you them all, and you can check them in the paper. Here, I will just show you one of the cell type specific ground truths from the genome-wide perturb-seq kind of data set, where you use perturb-seq or CRISPR-Cas9 to remove genes from a cell, deactivate it basically, and assess how this is changing the cell's expression after some time. And basically the genes that are changing in expression after you've removed one gene, you just mark them as connected, and that's how you define your network. And there you have a network that is really big, 8,000 by 8,000 genes, so that's really large. And that's also cell type specific to K562, which is a cell line. Again, that's SPRINT, and the other models have not seen during training. And on there, I'll show you both AUPRC, which you see is low, and EPR. And again, Gene3 performs really well. I haven't talked to you about DeepSem. You've seen the performance, which was on average not amazing, but it's kind of an autoencoder model, like a VAE, which learns, in addition to its weights, it's learned an interaction matrix between its input elements, so between genes. And there, scGPT performs better than GeneFormer v2. But SPRINT again outperformed the two foundation models and has learned likely a bit of a better gene networks. And what's super interesting is, and there is a good reason why, SPRINT, where you only use the heads selected by OmniPath, performs like almost random, or random to this benchmark. And the reason is if you just compare the OmniPath ground truth to the genome-wide perturb-seq ground truth, the overlap is also tiny and it's almost random. So meaning those two definitions of what is a gene network are very, very different to maybe what you were thinking, Denis. However, now if you select some heads that seem to correlate with genome-wide perturb-seq, you see that there is at least a set of heads in SPRINT that when averaged, when used, only those, they contain a lot of information about the genome-wide perturb-seq ground truth, which again I think is interesting and shows that this model have learned something about about the relationship between genes just from being pre-trained on those tasks. Okay, and maybe the last thing I want to mention is that genome-wide perturbs, selecting heads is not something that you can apply just to SPRINT. You could also do it with GeneFormer v2 and a CGPT. It would be quite some work to make this work, and just being able to rerun those two tools was a bit of work, and to extract gene network from those as well. I won't, because of time, I won't talk about the study that we did on benign prostatic hyperplasia, which is an early form of prostate cancer, where you can basically see that SPRINT can be used as an integral part of your bioinformatic pipeline. Generate prediction on cell type, embed your cells into a latent space, get at very specific subcell types, where actually you see that you get all of the known, if you do differential expression between your predicted cell types, you get a lot of the known markers that are actually associated with cancer or not for the different subsets. That you can apply denoising to identify new markers, and that you can also generate gene networks from which that are cell type specific. For example, here in the context of fibroblasts that are like normal fibroblasts and the ones that are associated with cancer, and see that you get hub genes and clusters that are also called communities that contain things that you would expect from fibroblasts that are in the context of prostate cancer in the stage of fibrosis, and generating a lot of extracellular matrices and so on. So you can read more about this in the paper. I don't want to take too much of your time. And conclusion is that I've mentioned that already, but a lot of work remains basically. There are still many issues both in SPRINT and in the other models. I hope that just SPRINT brings a lot of new ideas and also improved performance to these types of foundation models. But still, lots of ideas remains. The data itself is poor. You don't access, for example, non-coding RNAs, which for gene network or for gene regulation is really important. But yeah, I think SPRINT can still do quite well. Here you have the main figure, or kind of beautiful figure that just shows SPRINT's embedding on 2 million randomly selected cells from cell by genes, where you can see that actually the cluster make a lot of sense, and it removes a lot of quite some batch effects, although you still have some. And you can see that cluster with assess, you'll get known cell types, and from those clusters or those cell types you can generate cell type specific gene network. And this tool and all of those packages are completely, I hope, completely reproducible. I'm there on GitHub if there's any issue, and you can retrain from scratch SPRINT. You can do whatever you want with it. It's MIT license, and you can also benchmark your gene regulatory network inference tools on BenGRN and use the STL data loader. So yeah, thanks for listening and for the amazing people in my labs. And if you have a bit of time, I can answer some questions. I don't know if that's
Yeah, I would really love, if you allow me, I would really love you to talk a little bit about ablations, because you have a lot of like different elements of inspiration added on top of the base model, and it's really interesting to see which ones play the biggest role. For example, ECM encoding, basically taking this pre-encoding for genes, then different auxiliary losses that we are using, and perhaps how you normalize expression. I don't know. Okay, the last part, let's skip.
But so yes, you're completely right. We really need. So I haven't mentioned the ablation studies. Also, there are big models. So running ablation studies is heavy computationally, but I've run quite some, and you'll see the results in the paper for many ablations. Also, there is different sizes of SPRINTs that you can use, from small SPRINTs to like large ones. And you can get in the paper for the results. What happens is oftentimes it's not, especially if you use many different metrics, it won't be a clear-cut winner all the time. So there are moments where, for example, using gene location in the genome is really useful. For example, for denoising, it's really useful. For gene network, in the end, it's not that useful. It doesn't improve the performance at all, for example, to use gene location in the genome, but it improves the performance in predicting gene expression itself. for example, denoising itself is bringing a new ability to sprint. And as of now, we haven't run all of the possible benchmarks, and that will come in the final version of the manuscript, in the published version. But it seems that it brings this new possibility, but in gene network inference, compared to doing a hard mask language modeling of sprint, which you can also do, you can retrain sprint with mask language modeling instead, or doing both, also mask language modeling and denoising. Just doing mask language modeling also brings you quite of a good gene network. And so, there's a lot of ablation studies we've run. You can see the results. SC data loader, for example, allows you to run much better performance on cell type prediction, but we haven't assessed it well, I think. But if you just look at base loss curves, you don't see that SC data loader changes much the results. But I think that's because you'd want to look at rare cells mostly. Yeah.
Okay, thank you. I have 2 other questions, but I will let others ask first.
Yeah, let's maybe go with, can you comment on how the ESM2 embeddings of several isoforms are projected to the genomic locus? In that case, you should have a 1, the locus, to many, the embeddings of the isoform relation.
Yeah, but it's easy. There is no isoforms. Just most of the single-cell RNA-seq data don't have isoform level information. So here, the only thing that I use is the most common protein product. So kind of the landmark, most associated protein. If you go on ensemble, you'll get, a protein associated with that gene, basically. So clearly, it's not perfect, and there might be better ways. Actually, before I got dragged into other things, I was working in using more of a DNA language models embedding and really using the RNA or DNA sequence of the gene. Maybe that works better. Yeah, trying that out. But for now, it's really the protein product. And the idea is, first thing is you want some information about the protein, and for sure there will be splicing isoforms that might be more expressed. You just don't have this info in RNA-seq data most of the time.
All right, then Denise, do you want to go with
yeah.
your other points?
Yeah, so one question is, so you're using right now a hierarchical classification scheme to output a lot of different phenotypic traits of the sample. However, if you add things like cell perturbation studies, it will be a little bit hard to encode everything in that way. Well, like there are still possibilities to do expanding hierarchical classification and so on, but maybe you also thought about the way to output basically an embedding vector in a multimodal setting where you can essentially prompt your system by an embedding vector that encodes a perturbation, maybe even a small molecule added to cell culture, and maybe outputs also some general purpose encoding vectors that can be passed to other models, not just on expression and on expression, I think.
Yeah, well, that's a great question. Of course, I thought about this, although at some point you had to reduce the scope of what would be this project. Otherwise, my PhD would be really, really long. There might be some ideas for the next project, and I think a lot of people are working on this also, encoding perturbation. I don't think it would be too hard to get some embedding of a molecule or of a gene that you add, as an input to SPRINT to say, okay, now predict the perturbation effect of that. Just I haven't done it, but I'd very welcome any work on that for sure. And I think at some point you talk about reusing embeddings or scales of embeddings. And to me, that's something that is quite interesting and important where, what you see here is that, SPRINT tries to kind of have this model of the cell, right? As well as it can at least. And it's using for that another model's, embedding that was trying to have a model of the proteins and how they fold, right? This kind of ESM2. And so, you have this scale of the molecules that ESM2 takes and generates an embedding of the atoms and generates an embedding of the proteins. SPRINT takes this embedding of the proteins and generates one of the cell. And then now you have, those kind of spatial omics data where you could, maybe use those embeddings to generate a tissue level, more like a model. That to me is also kind of this idea of doing multi-scale learning and learning at different scale with different models.
Yeah, it's connecting with bulk expression is also probably useful because bulk expression is very ubiquitous. Yeah, and another question that I have is the most common question probably in CompBio. Have you tried to do like very strict validation where you actually try to remove from the training set like a whole family of cells, not just a single cell type, but basically like ablate a very large portion of the human physiology and see if the model understands something in that way, not just by interpolation, but essentially.
Are you doing analysis back with a model that maybe has just in one tissue, cells from one tissue type? That's what you meant?
Yeah, essentially something like removing all T cells, like every subtype of T cells, but retaining other lymphoid cells and looking if the model is able to recover things like protein-protein interactions or co-expression networks for the T cells by, yeah.
Huh, okay.
Essentially generalizing knowledge from other cell types into an obfuscated cell type.
I think, yeah, so I think it's really important to be able to do that. It's hard to know how, well, like what you would try to predict. Like you can try to predict just can it do denoising again for those cell types
Yeah.
seen.
Even
in a way, it's what I do for using a test data set. So it's maybe it has seen some of those cell types, but it hasn't seen basically this specific data set itself. But yeah, I think it's something I could try actually, like just not ablating data sets and having test and train data sets, but doing it even harder at the level of the cell type.
yeah, I think one way to increase the signal a little bit is instead of tracking the denoising loss on the entire reconstructed set of genes, prioritize genes that are known to be like either marker genes for example, in our example, T cells, that, for example, separate different variations of T cells, like different T cell helper families, like Tregs from non-Tregs, and essentially only focus on those cells, on those genes, and measure loss on them.
Yeah, actually, it's in my to-do list. This one,
Yeah.
like doing masking or denoising and looking at specific gene classes.
Yeah, because if the model is able to kind of unravel the fine structure of, let's say, T cell families, because there are a lot of different T cell variations, then it understands how internally these genes work and how you can actually impute them functionally. Yeah, and the last question, yeah, I guess I posted it, is this ERP loss, it's essentially precision recall loss on edges of the PPI network, right? So
Yes.
maybe it's interesting to track different types of PPI network edges, not PPI, but co-expression network, sorry. Because obviously some edges are just correlation edges, but other edges, they can be signed, meaning that, like, in the simplest case, we can have a transition factor that either increases or decreases the expression of the gene. And probably, I don't know, probably you can also like track the sign of the edge as well, and maybe just see what kind of edges are easy to recover and what kind of edges are hard to recover, because again, like predicting just a blob of just the giant component of the co-expression matrix, it's not super difficult because a lot of genes are very public, they have very high degree, and those edges are kind of trivial to predict. But some edges are very functionally important, and we probably want to concentrate on them.
Yeah, it's a good point. So I think at first, for the way, like the signed edges, there is a tiny amount in, for example, OmniPath. I couldn't even get it for, like, it would have been super hard to get it for genome-wide perturb-seq. Basically, they had only released the p-values and not the signs. So that's unfortunate. Also, as is, if you just take the average of attention matrices, you have values between 0 and 1, so you don't have a sign. Now you have some other attention mechanism where you can sign your attention, but it's a completely different model.
Actually, I think this is
Okay.
a good point. And last thing, just in this group, we discussed a lot of papers that actually solve this. Essentially, modification of transformer with something like sign positional embedding, like magnetic Laplacian or things like that. So essentially, you can make sign versions. But that's all, I think. Thank you.
Thanks.
Okay, so we're also done with your questions. Well, then we can call it a day in general. But Jeremy, any last words from your side?
No, yeah, just thanks, Hannes, for welcoming me in this to present my work, and thanks for everyone for listening. And I'm open to collaborations, to discussing projects. So I don't know if you have my email address, but I think Hannes I can send it to Hannes or put it on the Slack channel. And feel free
Okay,
to.
well, if you're on the Slack, then people can also just search your name and message you.
That works as well. Cool. Thanks, everyone.
Yeah, thanks everyone, and thanks for the question, especially Denise and also Peter, Anush, Francesco, and everyone else. And see you around, friends of the sun.